The Brainstem Navigator, a toolkit to study brainstem nuclei structure and function in living humans, by Bianciardi, Cauzzo, Toschi, Colli, Singh, and Garcia Gomar. I have no conflicts to declare. The brainstem is a stem-like area at the base of the brain. In humans, it's slightly bigger than a thumb, yet it contains several gray matter regions called brainstem nuclei, which are involved in vital functions such as arousal, sleep, motor, sensory, autonomic, limbic, and nociceptive function. However, due to their small size and limited MRI contrast in the brainstem compared to the cortex, these nuclei are difficult to visualize in conventional imaging of living humans, and extrapolations from 2D postmortem brainstem atlases are often used to identify their location in vivo MRI with limited precision. Our goal was to generate and publicly release the Brainstem Navigator toolkit that includes a deformable 3D atlas of 31 brainstem nuclei by 7 Tesla multi-contrast MRI and a tutorial of coregistration routines for precise alignment of the brainstem nuclei atlas labels to conventional and advanced diffusion functional and structural MRI. In 12 subjects, we acquired multi-contrast 1.1 mm isotropic resolution ecoplanar images at 7 Tesla, including a T2 weighted image and diffusion weighted images, from which we computed the fractional isotropy, and which enabled the visualization of brainstem nuclei on a single subject level, as shown here. After preprocessing, we performed manual and/or semi-automatic segmentation of brainstem nuclei, and then precisely registered both the images and the labels to stereotactic space. Spatial overlap across subjects was then computed to yield probabilistic atlas labels of 31 brainstem nuclei. The nuclei atlas labels were validated by inter-rater agreement, internal consistency, volume comparison to postmortem literature, and histology. Of the 31 brainstem nuclei, 16 were arousal and motor brainstem nuclei, such as raphe nuclei, reticular formation nuclei, cholinergic nuclei, noradrenergic nuclei, and other motor and arousal nuclei. And the other 50 nuclei are relevant in autonomic, limbic, and nociceptive functions, as well as in sensory vestibular functions. Delineations were performed by 2 postdocs in my lab at that time, Guadalupe Garcia Gomar and Kavita Singh, and by myself, across 6 years, and this work led to the 6 publications listed below on this slide. We packaged and recently released the atlas within the Brainstem Navigator toolkit, which you can find just by searching on Google the keywords Brainstem Navigator. After you download it, you will find the probabilistic atlas labels with a range from 0 to 1, but we also released the thresholded probabilistic labels with a range between 0.35 and 1, and the thresholded and binarized labels as well. There is a total of 31 nuclei, but if you count left and right nuclei and subnuclei, we have a total of 76 labels and files. We also delineated the 3 diencephalic nuclei, such as the lateral and medial geniculate nuclei and the subthalamic nucleus, which can be found in this folder. The atlas was developed in both 1 mm isotropic resolution Illinois Institute of Technology IIT stereotactic space to facilitate its use in diffusion MRI studies, and in Montreal Neurological Institute MNI stereotactic space for functional and structural MRI studies. To align users' diffusion data to the Brainstem Nuclei Atlas, we developed high-dimensional diffeomorphic bivariate nonlinear registrations of native fractional isotropy T2 weighted images to IIT templates. Note that the non-diffusion weighted B0 image can be used as a T2 weighted image. These routines were developed with my team and collaborators shown here. A crucial step for precise brainstem coregistration is masking out of areas outside the brain, which might be present in native images and absent in stereotactic templates. This includes removing scalp and non-brain signals, manual editing of the brain mask, especially to remove areas anterior to the pons and the medulla, which include, for example, basilar vessels, and cropping the inferior slices covering the spinal cord. This is an example mask that we used for coregistration. You can run coregistrations either on a subject-by-subject basis, as just shown, or if you already acquired the data for the whole cohort, you can run the coregistration to the IIT space via an optimal group template that can be achieved by iterative registration and averaging. Here we show quality of registration for an example diffusion dataset, indicating good alignment for all subjects in the brainstem. To align functional MRI data to the brainstem nuclei atlas in MNI space, first a subject-by-subject boundary-based affine or nonlinear transformation to the anatomical T1-weighted MRI is computed, and then this is combined with a nonlinear transformation between the native T1-weighted MRI and the T1-weighted MNI template. Also here, masking might be beneficial. We show the coregistration quality for an example functional MRI dataset, indicating a good alignment for all subjects in the brainstem, except for some residual distortions and signal dropout in some subjects in the anterior part of the pons, which likely is an area where we do not have any brainstem nuclei atlas labels. Here we show example application of the Brainstem Navigator toolkit to investigate brainstem hodology in health. We applied the atlas and coregistration routines to 7 Tesla functional and diffusion data acquired on 20 healthy subjects, and computed the resting state functional MRI connectivity and diffusion-based tractography for 31 brainstem nuclei towards a parcellation of the whole brain. We found a significant correlation between functional and structural connectivity matrices. Specifically, we observed a similar stronger connectivity of brainstem nuclei with the frontal cortex and subcortical regions, and weaker connectivity with the visual cortex, although with some differences, as the structural connectome was denser within the brainstem and the functional connectome was denser within the cortex, possibly due to the respective different sensitivities and biases of the two techniques, namely the higher sensitivity of fMRI connectivity towards larger regions and the tractography bias towards larger fiber bundles and fibers closer to the seed. We also verified the translatability of our 7 Tesla connectivity results to 3 Tesla MRI, and we found good association and a high number of common functional links between 7 Tesla and 3 Tesla MRI. Similar results were observed for the structural connectivity. Finally, we also recently provided a translational application of the Brainstem Navigator toolkit to investigate the location of microbleeds in traumatic coma at 3 Tesla, and to determine changes in the structural connectivity of brainstem nuclei at 7 Tesla in pre-manifest synucleinopathy. In summary, we developed, packaged, and released an atlas of 31 brainstem nuclei and 3 diencephalic nuclei, which has lower spatial resolution than postmortem atlases, yet it was developed in living humans, it's 3D, probabilistic, which means it accounts for variations across the population, and it's deformable, which means it can be automatically mapped to conventional 3 Tesla MRI of living humans. We also developed coregistration routines for conventional and advanced MRI, which we are currently improving by also incorporating users' feedback before we release them. We are currently developing a brainstem nuclei atlas in elderly humans. Check out this poster by my team this afternoon, and we hope this will be a useful tool. We would like to thank our collaborators, funding sources, and you for your attention.